Hello everybody and welcome back to my channel and welcome back to Biology with Gracie. So as you can see from the title of today's video, this is going to be part one of my immunology videos. It's going to be two parts, and this is specifically for Edexcel A and B A-level biology. However, if you're just interested in biology or you are on another spec, this is still very relevant. The specifications are pretty similar across the board. As you've probably seen, I've also got a previous video that I did about a year ago on immunology for all of the exam boards. So if you want to go check those out as well, then you can. They've got some really helpful exam practice at the end of each video, which you might find helpful. So I will link those down below and in the cards as well. But without further ado, let's get straight into the video. Okay, so we're quickly going to go over the content of today's video. So we're going to be talking about white blood cells, T cells and B cells, the humoral response, the cell-mediated response, and then we're going to finish talking about antigens and antibodies. So for Edexcel A, this is the specification points that we're going to cover today. So you've got 6.7, 6.8 and 6.9 for topic 6, which is immunity, infection and forensics. I'll give you a moment to pause the video and read through those if that's relevant for you. And then we've got our specification points for Edexcel B, so topic 6, points 1, 2 and 3. And again, I'll get you to pause the video and read through those if that's relevant for you. And so first we're going to start. Comment down below, how do you feel about immunology in A-level biology? Is it something that you struggle with? Is it something that you feel quite confident about? Let me know down below, and whilst you're there, make sure to subscribe. So first we're going to start with white blood cells. We're going to talk about the types, the structures and the functions. First we're going to start with our two types of white blood cells. So we've got phagocytes and we've got lymphocytes. These are the main two groups. We've got macrophages, B cells, T cells, neutrophils and monocytes. These are all different types of white blood cells that fit into the two categories of phagocytes and lymphocytes. So pause the video now and see if you can work out which of these white blood cells in green match up with phagocytes, or they are phagocytes, and which are lymphocytes. These are the correct answers. So we've got neutrophils, monocytes and macrophages. They go with phagocytes, and B cells and T cells are lymphocytes. So well done if you got that correct. If not, don't worry, now you know. We're going to talk about white blood cell functions. So we've got phagocytes and lymphocytes again, and then we've this time got mast cells and other white blood cells, and we've got eosinophils. So I'm going to read out some statements now, see if you can match them up to the correct group. So we've got the first statement. So which white blood cells are involved in the immune response, including antibody production and immunity. Then got produce chemical histamine involved in the inflammatory response. Respond to parasitic infections by producing cytokines which activate other white blood cells known as leukocytes. And then we've got neutrophils and monocytes leave blood capillaries by squeezing through cells or capillary walls. Neutrophils ingest and destroy bacteria. Monocytes become macrophages and engulf bacteria, foreign matter and cell debris. So see if you can match those statements or those functions up with the correct white blood cell group. Now we're going to go on to the answers. So phagocytes matches up with the statement about neutrophils, monocytes and macrophages. These are all, of course, phagocytes, and this is the process of how each of these works within the immune system. Lymphocytes, so we know that they're involved in the immune response, including antibody production and immunity, so B cells being key to that antibody production. Mast cells are the ones that produce histamine, which is involved in the inflammatory response, so the non-specific immune response. And then finally eosinophils, so these are the ones that respond to parasitic infections by producing cytokines. So they activate other white blood cells such as leukocytes. Now we're going to go on to white blood cell structures. So here we've got 4 different types of blood cell, and then again we've got 4 different statements describing the structures. So we've got an eosinophil, which has a lobe nucleus, usually 2 lobes and granules in the cytoplasm that may be stained pink. Monocyte, largest white blood cell, often with kidney-shaped nucleus. Lymphocyte, the nucleus usually fills most of the cell. And then we've got a neutrophil, so this has a lobe nucleus, often 3 to 5 lobes, and granules in the cytoplasm that may be stained light pink. So see if you can match those up with the images on the left. Okay, so hopefully you didn't have too much trouble going through that, but don't worry, we're going to go through the answers now. So the first white blood cell that we've got at the top on the left is actually a monocyte. So this is the largest white blood cell, often with a kidney-shaped nucleus. So you can see that sort of kidney shape within the monocyte here. So that is the kidney-shaped nucleus. So that's something to look out for if you ever get a question like that. We've then got the neutrophil. So this one, remember, has a lobe nucleus of usually 3 to 5 lobes. So you can see here, this purple, this is the nucleus, and this is actually split into about 6 here, but it's usually about 3 to 5, and that's usually what gives it away for being a neutrophil. Something else to look out for as well is the granules in the cytoplasms, which may be stained like pink. So you can see these granules here, these dots around the neutrophil. We've then got the lymphocyte. So this states that the nucleus fills most of the cell. So we've got a big nucleus here. I've seen in exam questions before that the nucleus actually is even bigger than in the image here, and it just looks like a black circle, the whole cell. You can't differentiate the nucleus essentially from the side of the cell. So that's something to look out for too. And then finally, we've got our eosinophil. So this also has a lobe nucleus like a neutrophil, but it usually only has 2 lobes. And it has granules as well that may be stained pink, similar to the neutrophil. One thing I've noticed is that some people get a bit confused between the monocyte and the eosinophil, purely because these 2 lobes, it can sort of look like a kidney shape, but I think it's just key to remember that this is definitely one shape. Whereas this is sort of split more into 2, if that kind of makes sense. So now we're going to move on to T cells and B cells, which are also known as T lymphocytes and B lymphocytes. So first we're going to discuss types of T cell. So before I go through them, see if you can remember them, write them down, pause the video and try and remember what each of these T cells are. Okay, so they are in fact T helper cells, T killer cells and T memory cells. So again, we've got some statements to describe them that are jumbled up, so you can pause the video and actively try to match them up. So the first one of these cells attaches to antigens on a pathogen infected cell and kills the cell. The second one, so it remembers the pathogen specific antigen and recognises it a second time, and they divide into the correct type of T cell to kill the cell carrying the antigen. And then finally, one of these T cells releases substances to activate B cells, T killer cells and macrophages. So pause the video now and see if you can match up these statements with the correct T cell. Okay, so now we're going to go through the answers. So T helper cells, they release substances to activate B cells, T killer cells and macrophages. T killer cells are the cells that attach to antigens on a pathogen infected cell and kill the cell. And then T memory cells are the ones that remember the pathogen-specific antigen and recognise it that second time around, and they're able to divide into the correct type of T cell to kill the cell carrying the antigen. Next up, we've got B cells. So again, see if you can remember what they are before I go through them. But the first B cell is in fact B effector cells, and then we've got B memory cells. So one of these is a longer lived cell, remains for months or years in the body, and it enables that individual to respond more quickly to the same antigen in the future. And then the other one differentiates to produce plasma cells, which release antibodies into the blood and the lymph. And these cells are usually relatively short-lived, lasting only a few days compared to the longer other B cells. So see if you can work out which B cells these statements match up with. So it is the B effector cells, which are the ones that differentiate into plasma cells, releasing antibodies into the blood and lymph. And then it's the B memory cells, which are the longer-lived cells, usually remaining in the body for months or years, which can enable that individual to respond more quickly to the same antigen in the future during a secondary immune response. So now we're going to go through the humoral response. So this involves T helper cell activation, clonal selection, and the roles of B cells. Okay, so T helper cell activation. So this is a key part of the humoral response. So the first stage in T helper cell activation is you are infected with a pathogen that has antigens on its surface. So then that pathogen is engulfed by our key white blood cell, our key phagocyte and macrophage. And then the macrophage actually presents the antigen on its surface. And as the name suggests, it becomes an antigen presenting cell. So then this allows the antigen presenting cell to bind with a T helper cell that has complementary CD4 receptors. So then the T helper cell becomes activated and it divides. And it divides into clones of active T helper cells as well as active T memory cells. Now we're going to discuss clonal selection. So the first stage again is we've got a pathogen with antigens on its surface, and the antigen binds to the B cell with a complementary receptor. So then the B cell becomes an antigen presenting cell. And then the activated T helper cell, so remember from our T helper cell activation portion of this video, with those complementary receptors binds to that antigen presenting cell and produces cytokines, which are a form of protein that stimulate that B cell. So then we're going to go on to the next slide to discuss the next few stages. From that, we actually get a clone of B memory cells and a clone of B effector cells. So these B effector cells differentiate into plasma cells, which secrete antibodies. So these antibodies are able to bind to the antigens and identify them for destruction. So now we're going to talk a little bit more about the role of B cells within the humoral response. So the B cells, as we said previously, secrete antibodies in response to antigens. The antibodies are then able to bind to the antigens and label them. So then the antibody binds to the antibody receptor. So this could be on a macrophage, for example. So then that macrophage engulfs the antibodies and the pathogen. And then lysosomes, which are a form of organelle within cells, are able to fuse with the vacuole, releasing digestive enzymes, and those are what destroy the pathogen. Okay, so that was the humoral response. I know that it's fairly confusing. That's why I've tried to set it out into 3 simple stages rather than all in one go, compared to what a lot of textbooks and revision guides do, because I know that a lot of exam questions ask you about specific sections such as T helper cell activation or the role of B cells, so that's why I've separated those up. If I went through that too quickly, or you think you need to look over it again, then make sure to head over to my website where you will find all of these resources, so the PowerPoint presentation, so that you can go through them in your own time, at your own speed, and have a look in more detail at those diagrams. But anyway, we're going to go on to the cell-mediated response. So this mainly involves the T killer cell. The role of the T killer cell. So T killer cells have complementary receptors which bind the antigen presented on the body cell. And then these T killer cells divide to form active clones and T killer memory cell clones as well. So the division is stimulated by cytokines from T helper cells, which we talked about previously. And the reason that these cytokines are really important is that there wouldn't be enough T killer cells produced to fight that viral infection. So it's really important that those cytokines are produced by those T helper cells, that they can stimulate those T killer cells to divide, so that more are produced, so that they can kill more of that infectious particles. Then the next stage is the T killer cells releasing enzymes, and these enzymes create pores in the membrane of the infected cell. This means that ions and water can flow in and the cell can undergo lysis, and lysis is the process of the cell swelling and bursting. And then the pathogens within the cell are released and labelled as targets for destruction by antibodies from B cells. So, following on from that, we're now going to talk about antigens and antibodies and how we can identify pathogens via their antigens and how we can destroy them through antibodies. Each type of cell is identified by a specific molecule, and this specific molecule, which is actually a form of protein, is known as an antigen, and these are found on the surface of cells. Antigens are forms of proteins, and they also can be glycoproteins, and each protein may have a number of antigenic regions, and a whole organism, such as a bacteria, will carry many antigens. So essentially, the antigen is the part of the organism or the substance which is recognized as foreign by the immune system. It essentially causes that immune response. Okay, so that was antigens. So now we're going to talk about antibodies, and we're going to talk a little bit about their structure. So antibodies, like antigens, are proteins also, and they're made up of 4 polypeptide chains. So we've got 2 heavy chains and 2 light chains. And when an antigen binds to the antibody, it's known as an antigen-antibody complex. So antibodies also have a constant region. They have a variable region. So the variable region, it's the binding site, which is complementary to the antigen. So it has a specific amino acid sequence, and hence the shape of the binding site is different in each type of antibody. It essentially changes for each antigen. So you may be thinking a little bit about this and think about how that links to another topic. So for example, enzymes, they have an active site which is specific for a specific substrate. So it's similar here that how a specific antibody fits a specific antigen. And then they have a hinge region, and this is what allows flexibility when the antibody binds to the antigen. So as you can see here, we've got the antibody structure. We've got those antigen binding sites at the top here in pink, and then we've got the 2 light chains, so one here and one here. And then we've got the heavy chains at the bottom, and then these little divots in here are the hinge regions. So antibodies, as I said previously, are protein molecules, and they're actually of a class known as immunoglobulins. So these little rectangles in purple are disulfide bonds. The blue represents the constant region, and the pink represents the variable region. So you may be presented with this structure in an exam, or you may be asked to draw it yourself. So it's really important that you remember this structure of an antibody and what each of the parts are important for, such as the hinge regions for flexibility, and then the 2 light chains, 2 heavy chains, and then the specificity of antigen binding sites. In terms of antibody production, so we know that B cells are our immune cells that produce antibodies, and each B cell actually produces only one type of antibody, as we said previously, which binds to only one specific antigen. So as we said before, a microbe could have several different types of antigen on its surface, but each different antigen will bind and activate different B cells. And B cells have receptors on their surface, which include transmembrane versions of the antibody molecules that they produce. Antibodies have 2 forms. So we've got the membrane-bound form. So this is the antibody which is attached to the membrane of a B cell. And then we've got a secreted form. So this is free from any attachment. So these 2 forms actually have slightly different heavy chain proteins. One of the reasons for this is that the membrane-bound antibodies actually have an extra section of protein, and they need that to anchor them to the B cell membrane. But since the secreted antibodies don't attach to the membrane, they're free from any attachment, they don't need that extra bit of protein. So that is why there is a difference between those heavy chain proteins on those different types of antibodies. Finally, just a little bit more information on antibodies. So both heavy chain proteins are actually coded for by a single gene, and this is copied into mRNA for protein synthesis. So there's a bit of a crossover between topics here. If you don't know, mRNA is a type of RNA called messenger RNA. If you do want to find out more about that, then go and have a look at my protein synthesis videos, and they will explain everything to you and explain this slide again in a bit more detail. But it's possible to actually create more than one protein from the heavy chain gene, and this is done by modifying the mRNA before it's translated into the protein. So it's actually known as a post-transcriptional modification. And again, if you want to find out more about that, then feel free to go and have a look at my protein synthesis videos. So that is our first part of my 2 immunology videos done. Make sure to comment down below, how do you feel about immunology now? Maybe you felt a little bit confused about it at the beginning, that's why you wanted to watch this video, and hopefully you feel a bit more confident now. If you would like to, then you can use one of these emojis to describe how you feel. So if it's a very useful video, you feel like you've learned a lot, then you can use the starry-eyed emoji. If it was quite a useful video, you think you learned some things, then you can use the smiley emoji. If you found it very enjoyable and engaging as well, then feel free to use the little blushing emoji. And if you want more videos like this, so that you can learn more, then feel free to use the little nerd emoji. You can use more than one of these to describe how you feel. And if you want to write a longer comment, that would absolutely be great as well. Thank you so much for watching this video. Don't forget to subscribe down below. It's free, it's easy, it supports me and the channel, and it also builds a bigger community of science-loving students. But if you do have any more questions, then feel free to comment them down below or message me on Instagram at Biology with Gracie, and I will answer those as soon as I can. But I do hope you enjoyed this video. Again, make sure to subscribe, leave any comments down below, and I will see you in my next video. Bye guys.